Welcome to the IGVF Scientific Seminar Series. I think today we will hear two presentations, one from Jason Ernst and another from Vikram Agarwal. I believe that the latest format is to give 20 minutes presentation and then 10 minutes for discussion. So why don't we start with Jason with computational approaches for large-scale modeling and analysis of epigenomic data across diverse tissue types and species.
Okay, so thank you for the presentation. Giving me this opportunity to present here. So as Ian said, I'm Jason Ernst, and I'm going to be giving sort of a PI type talk and try to give an overview of three methods that came out of our group sort of recently. And this was a talk I was planning to give last year, but because of scheduling reasons, I was brought to this year. So to talk about the first topic, the large-scale chromatin state modeling. So what this is motivated by is chroma HMM annotations and reference epigenome data in general has been useful for a variety of tasks for understanding the non-coding genome and non-coding variants. So what you're seeing at the bottom here are maps of different histone modifications, and I previously developed methods which I assume many are familiar with, including chroma-HMM, which can take multiple different histone modifications and annotate the genome based on their combinatorial and spatial patterns into different types of genomic elements, including different types of promoters and enhancers. And what you're seeing here is a chroma-HMM annotation across 127 reference epigenomes, which was part of the Roadmap Epigenomics Consortium effort. And some of the input data to this was imputed based on chroma-impute, which can do an imputation. But one of the challenges and opportunities now is we now have this type of reference epigenome data for hundreds, and soon will be close to 1,700 reference epigenomes. I'm currently collaborating with the International Human Epigenome Consortium, which has recently finished the processing of data from about 10 different epigenome mapping projects and combined it in a unified way, which will have 5,000 ChIP-seq data, and we've imputed more than 4,000 data sets that are only available in imputation format, and we're in the process of generating chroma-HMM annotations for 1,700 reference epigenomes. But this sort of presents an opportunity, having all this sort of diverse data, but challenges in terms of how does one actually work with so many different data sets in a meaningful way. And I'm going to talk briefly about two strategies for this. And all the work I'm going to talk about is either published or on bioRxiv, so you can consult that for more details of the method or the evaluations. So one approach is to group samples based on known common biological properties and then come up with a summary annotation. And a PhD student in my group, Havu, developed a method which would estimate the probability that any location is in a specific chromatin state using multi-class logistic regression classifiers. An ensemble of them were trained to predict the chromatin state in each sample given other samples in the group. So this allows one to get a probabilistic estimate of the state assignment at any location, taking into account the variability of the state assignment at that location and the relationship between states across the genome. And then you can give a hard assignment of the chromatin state annotation, as well as use the probabilistic estimates for other applications, for example, to compare two groups of samples to identify chromatin state changes. But there's another modeling approach that has advantages in this context where you have many cell and tissue types. And this was a figure from a previous tutorial or protocols paper I'd written on chromHMM, that traditionally chromHMM in the multi-cell type context is applied where you have one common model, but then cell type specific state annotations. But an alternative approach is you have what we call the stacked approach, where all the data is inputted into the model at once, and you're learning a single annotation of the genome, and you're increasing the complexity of the state definitions. But this has some complementary advantages. It's not replacing the concatenated modeling, but it has advantages in that it can simplify the annotation output. So instead of, in the case if we have 1,700 reference epigenomes, having to deal with a single, 1,700 state assignments for any given location, we have one state assignment per location. And then these state definitions can differentiate constitutive from cell types specific activity. And it can also bypass the need to pick one cell type or consider all of them, because for many applications, one isn't necessarily interested in a specific cell type. For example, if you're studying a sequence variant and you don't know the cell type that's relevant, it's not natural to just pick one cell type and work with it. And then it also leverages more data simultaneously, which can improve some annotation. So this is showing the application of this modeling approach to the roadmap epigenomics compendium. So the rows are states, the columns are different experiments color-coded by the mark and the cellular tissue type group. And this is work that, again, was led by Havu and is published if you want more details. But this is showing the mission matrix and now color-coded by the top 10 omitted marks. And then we can also show the cell type groups of these top 10 marks. And just to give an example here, this state here is associated with what we characterize as enhancers that are specific to embryonic stem cells or iPS cells. And what might not have been expected is that we can also find a state here which is associated specifically with H3K9ME3 heterochromatin in ES or IPS cells. And H3K9ME3 is a more constitutive mark, so finding a state that's specifically associated with this group of samples in K9ME3 is something that sort of came out of this modeling of all the samples together. And there's many other sort of interesting states that one is able to infer by looking at all the data at once. And just to point out, like, one application that might be relevant to some of the IGVF efforts is there's a lot of variant prioritization scores that provide one score for the entire genome. And it's not always sort of clear how these different scores differ from each other. But by having a single annotation for the genome that's not specific to the cell type, we could start computing enrichments for the bases prioritized by these different scores. And what I'm showing you here are the enrichment for top 1% non-coding bases prioritized by each score, and then the fold enrichment. And you can see different scores prioritize different classes of bases, and it's color-coded based on the top 5 enrichments for each state. So for example, one can see a set of scores that are most enriched for what we characterize as certain types of enhancers, while not necessarily that enriched in promoter regions, while there's other scores that are heavily enriched in promoter regions, and in some cases even depleted in enhancer states. So then this is sort of already gives you a first order difference understanding of what the types of bases various scores are prioritizing. And we've also now done this for the mouse as well as the human and have an analogous universal chromatin state annotation for the mouse. Now, moving into the second part about gene-based epigenomic modeling. So it's quite common for people to be applying chromHMM, but then they're, at the end of the day, actually interested in doing some gene-based analyses. And you can see across a given gene that you'll have like different chromatin states depending on where you are along the gene. And sometimes there's various post hoc methods to associate a chromatin state with a gene. For example, one simple thing is to take the chromatin state of the transcription start site, but that would like lose a lot of information. So what we sought out to do was to have a principled model-based approach to annotate genes based on the full set of epigenomic annotations across, data sets across the gene body and 2kb flanking regions. So what we're doing here is effectively, these are 3 different genes, and we're giving them a label of a different gene-based chromatin annotation based on the entire set of data across the gene that's learned de novo. And the strategy that we developed for this, and this was led by a former graduate student in my group, Arthur, was based on a mixture of hidden Markov models. So the idea is that the set of marks that are found across a gene and its flanking region is modeled by one of k number of hidden Markov models. And these hidden Markov models have a relatively limited number of states. We have 3 states per hidden Markov model. And then we're going to learn the parameters of these hidden Markov models de novo from the data, and then assign each gene to its most likely mixture component. And the emissions are modeled the same way as chromHMM, based on a product of independent Bernoulli random variables. And this approach is built on top of chromHMM. So this is showing an example here where each of the big 3 groups correspond to a different mixture component, and then each of the individual rows correspond to one of the substates. And then we've given biological descriptions associated with these different states. Oops. And then what you're seeing here is an assignment grid. The rows correspond to genes, and the columns the different cellular tissue types. And just to give an example of one analysis here, we can compare these annotations to gene expression and high PLI genes. So the rows correspond, the x-axis corresponds to gene expression, and then the dots correspond to a combination of cellular tissue type and gene annotation. And then the y-axis is the proportion that's considered a high probability of being loss of function intolerant, greater than 0.9. And what you're seeing here is that there's a wide variability in the expression of the gene depending on the chrome gene annotation, and then also as well with the high PLI. And then importantly, even for annotations that have roughly similar PLIs or gene expression score, you can see variation in the proportion of high PLI genes. So this is saying that these annotations can capture additional information that's biologically relevant about genes that aren't necessarily captured at the gene expression level. So you can have genes that have, for example, are unexpressed, but are marked with different sets of epigenomic annotations that can correspond to different biological properties. And then the last part here is looking at human mouse epigenome conservation. So the idea is that it's useful to know if some location in human might have a similar role in mouse. And particularly, we're focused primarily on the 40% of the human genome that lines at the sequence level, but we would want some additional evidence that it might have a similar role in mouse. And the idea is to leverage the large compendium of data available in human and mouse, and to sort of leverage all this data at once, not just focusing on matching a single cell type or a single assay. So a former PhD student in my group, Subin Kwan, developed a method that could integrate thousands of data sets from human and mouse to come up with a single score of evidence of conservation. And some of the key features of this method is it does this by integrating different types of annotations without explicitly matching the annotations by their biological origin. And we're providing evidence of conservation at the functional genomics level. We don't necessarily capture, like if regions only active and we only have data from one of the species that's relevant, we might not call it as have evidence. So we're only sort of confident in things that we give high scores to, in a sense. So briefly, how this method works is we train an ensemble of neural networks to discriminate locations that align to each other at the sequence level as positive pairs, and the features are just based on epigenomic or functional genomic annotations, and then the negative pairs are shuffles of the positive pair. So these are locations in human and mouse that align somewhere to the other species, but not to each other. And then the neural network sort of learns a mapping to discriminate pairs that are true sequence alignments from shuffles. And then we would then go and apply this to a new pair. So the assumption here is that pairs that align at the sequence level are more likely to have a similar role, but we're not assuming that all of them have to be. So we applied it to a large set of data, more than 8,000 data sets in human, 3,000 in mouse. And this is just an example of a location. Boxes are showing roughly sequence aligned positions, and then the track is showing the score it gets. And you can see this is a chromatin state model that was learned jointly on the two species, that you have sort of more similar annotations where it got a high score. And then we could also understand sort of the conservation of different chromatin states as a function of the score between species. This is the annotations of the states defined in human. So just to summarize, and I'm about out of time, I presented a few methods, the CS-rep method for chromatin state summarization, universal chromHMM annotations in the human and mouse, chromgene for gene-based epigenome annotation, and LeCif for human mouse epigenome conservation, learning evidence of that. And I included links to the papers and preprints, and also GitHub links associated with them. And I'd like to acknowledge the people involved in the work, and I put in bold the people who did most of the work I talked about. So thank you for your attention.